Molecular biology of the gene, chapter 12, 5 sections: the genetic material, replication of DNA, the genetic code of life, and then looking at protein synthesis, the 2 steps, transcription and translation. So basically the genetic basis of eye, hair, and skin coloration. We know that all life on Earth contains the 4 bases of DNA, A, G, C, and T. And these bases are molecules and code for biological parts, such as the proteins that make skin, bones, and eyes. Different combinations of these bases are what makes up our genes. Remember that genes are functional units of DNA, and when we talk about it, the human genome itself, we're looking at somewhere around, I would say, 30,000 genes. The gene MC1R is a gene that contributes to skin, hair, and eye color in humans, and it is found in the nucleus of every cell. Some of the cells in skin do become specialized pigment-making cells that are termed melanocytes. Humans have variations in their MC1R gene that comes from their ancestry. So the differences in the DNA base sequence can alter gene expression, which affects how much melanin is going to be produced. Everyone has melanin genes, but each individual's gene expression is determined from the information in their DNA. And when we talk about that gene expression, we're looking at the phenotype, essentially. So the genetic material, the race for understanding what was the molecule of importance from passing on information from generation to generation. Is it RNA or is it DNA? Well, the following scientists are going to come to solve that answer. One of them would be a guy named Frederick Griffith. Basically investigated the virulence of Streptococcus pneumoniae, and he concluded that the virulence could be passed from a dead strain to a non-virulent living strain. That process itself is called transformation. So you do see a change there, or some type of transform in the bacteria. Further research by Avery discovered that DNA is the transforming substance, and DNA from the dead cells incorporated in the genome of the living cells themselves. So DNA and proteins were the candidates for the hereditary material. Basically, we know that proteins contain 20 amino acids that can be sequenced in different ways. The DNA and RNA each contain only 4 types of nucleotides. The requirements for the genetic material must be able to store genetic information, must be stable and able to replicate accurately during cell division and transmit it from generation to generation, and it also must be able to undergo mutations to provide genetic variability. So that is what scientists are looking for when they were trying to search for which molecule is responsible for doing this. And researchers showed that DNA can fulfill all these functions. So Griffith and his transformation experiment, basically what he did is mice were injected with 2 strains of pneumococcus, an encapsulated strain and a non-encapsulated strain. The encapsulated strain is the virulent strain, and the mice ended up dying. It has a mucus capsule that forms shiny colonies. The non-encapsulated strain forms dull colonies. So ultimately what he did is when he put in the encapsulated strain, when he put in the strain, the S strain, the mouse ended up dying. When he injected them with the R strain, the mouse ended up living. So Griffith then took the S strain and he heat shocked it. And what happened is when he injected that heat shock S strain into the mouse ended up living. But he then took that injected heat, heat shock strained, and mixed it in with a live culture of the non-pathogenic strain of the pneumococcus. And ultimately, one would suspect that the mouse should live because you heat shocked the pathogenic strain or the disease-causing strain, and you mixed it with the live non-pathogenic strain. But what ended up happening is the mouse died. So there was some sort of transformation there where there was a passing of genetic information from the pathogenic strain to the non-pathogenic strain that was alive that eventually led to the mouse death. So Avery, McLeod, and McCarty's experiment. Scientists argued that DNA lacked variability to be able to store that genetic information. However, Avery and his colleagues used enzymes that break down DNA, or DNase, or RNase, or protease, in separate experiments to digest the substance, which allowed for Streptococcus to produce a capsule and become virulent. The only enzyme that had an effect was the DNase. Ultimately, this prevented the transformation from occurring. These experiments show that DNA indeed was the transforming substance. It wasn't an RNA molecule and it wasn't a protein molecule. So Avery, McLeod, and McCarty started to come up with an answer to the question that many people were seeking. The other two, Hershey and Chase, they used phage, bacteriophages, to study whether it was DNA or RNA. The bacteriophage, remember, itself is a virus, and what they did is they hit it with radioactive material to label the DNA, and radioactive material to basically label the protein coat of the virus. And they used that virus, then, the bacteriophage, to infect E. coli bacteria. They discovered that the radioactively labeled DNA, but not protein, ended up inside the bacterial cells, causing them to become transformed. Only the genetic material could have caused this transformation, as the experiment showed that only DNA and not protein was the genetic material. So let's look at the genetic material today. I apologize, I was interrupted there. Transformation of organisms today. You hear about these a lot, genetically modified organisms, or GMOs. This is the result of the so-called transformation of DNA. Genetically modified organisms essentially are an invaluable tool in modern biotechnology today. Commercial products that are currently much used are genetically modified. One of those things that we can look at is the green fluorescent protein, or GFP, used as a marker. A jellyfish gene codes for GFP. The jellyfish gene is isolated and then transferred to a bacterium or the embryo of a plant, pig, or mouse. Ultimately, when this gene is transferred to other organisms, the organisms will then glow in the dark. So here you could see a picture of the experiment that was done. The bacteriophage right there. The phage looks pretty much like a foreign spaceship landing on the moon, but that structure there would actually be the E. coli cell. So this is the outer surface of that cell, and there it is injecting its viral DNA into that bacteria cell. So the genetic material itself. DNA contains 2 nucleotides with purine bases, adenine and guanine, and then 2 nucleotides with pyrimidine bases, thymine and cytosine. I always say that to remember which ones are purines and which ones are pyrimidines, I always think of agriculture, AG. You want pure agriculture, which would be adenine and guanine, purines, and then the other 2 pyrimidines, cytosine and thymine, abbreviated AGTC. Erwin Chargaff, related to the bases, he did a study, and from his study he said the amounts of ATCG in DNA are constant among members of the same species and vary from species to species. In each species, there are equal amounts of adenine and thymine and guanine and cytosine. All of this suggests that the DNA uses complementary base pairing to store genetic information. So when we think about it, each human chromosome contains, on average, about 140 million base pairs. So the number of possible nucleotide sequences is 4 to the 140 million. If you look at it, here you can see the DNA composition of various species. Humans at the top, Drosophila melanogaster, corn, fungus, and 2 strains of bacteria, Escherichia coli and Bacillus subtilis. Now, you could see the structure of the nucleotide bases there as well. The structure of each individual base is beyond the scope of the AP exam, but just to give you an idea of what they would look like, you could see it in the diagram there. Those that are purines and those that are pyrimidines. Coming closer to the structure of DNA, now that we know that this is the molecule of interest, there is a race to discover its structure and to provide more information on this DNA molecule. One of those pieces of evidence that helped greatly in the discovery of the structure of DNA was this evidence of X-ray diffraction. The X-ray diffraction was taken by Rosalind Franklin, who studied structure of DNA using X-rays. She used a crystallography technique. She found that if a concentrated viscous solution of DNA is made, it can be separated into fibers. Under the right conditions, the fibers can produce an X-ray diffraction pattern. She produced the X-ray diffraction photographs, and this provided evidence that DNA had the following features. 1, DNA is a helix, and 2, some portion of the helix is repeated. Ultimately, a colleague of Franklin's, Maurice Wilkins, showed her photograph to James Watson, who understood its significance about DNA structure. So there we see Rosalind Franklin and her X-ray diffraction there. And ultimately what we see is Watson and Crick, the model proposed in 1953 was a double helix model, is similar to a twisted ladder, where you have a sugar phosphate backbone that makes up the sides, hydrogen bonded bases make up the rungs, and the 2 DNA strands run anti-parallel to one another. Information is stored in DNA and must be read in the 5' to 3' direction. So DNA is always replicated in a 5' to 3' direction. And we'll look at that a little bit more in class. Complementary base pairing ensures that a purine is always bonded to a pyrimidine. So you have A with T and C with G. And for their research in the structure of the DNA molecule, they received a Nobel Prize in 1962, Watson and Crick. And here you can see the structure of the DNA molecule. One end, by meaning running anti-parallel, one end is 5' to 3'. The other end is 3' to 5'. So it's like they're running upside down to one another there. Okay, that's all for today. We'll get into replication and transcription and translation in our next lecture. Thank you and have a nice day.